Okay, thank you so much. Yesterday I was talking at the Audacious Goal session, and the modulator was talking about 15 years from now. So I said, no, 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 5 years. The reason is that 5 years ago, we didn't imagine next generation sequence. No next generation sequence was present 5 years ago. Also, 5 years ago, we didn't have the iPS cells. Also, we didn't have the CRISPR system to edit the genomes. We also didn't have these 7M8 vectors that can penetrate the whole layer of the retina for gene therapy. All this really advanced technology was not present 5 years ago. So 5 years, how much you can do. So for Audacious's goal, it's not 15 years, but it's 5 years, and we're going to finish this, at least the Mendelian part of this disease for all the genetic diseases. So I'd like to start with the what we have been doing for the last 4 years. This is about the 4 years project. And we have been studying these various retinal diseases. And as you know, there's a Stargardt and retinitis pigmentosa, Leber congenital amaurosis. These diseases are not stoppable, not curable, and in only just few cases with a gene therapy now, and it's very difficult. So we want to know first what is causing these diseases. And unfortunately for the Japanese population, most of the gene has been published is not actually found in the Japanese populations. So the past 25 years now, there has been a lot of genes mapped and also identified. However, and these are the genes that has been identified and then on the web from the retina network. However, if you look all this of the genes and then you try to diagnostic in the Japanese population, it's very difficult to find a mutation in these genes. That's why we started a consortium within Japan to tackle why is it not finding these mutations in the Japanese populations. So we assembled a consortium of 25 universities, ophthalmology departments from north to south, and most of the doctors are very knowledgeable for the ERG techniques to diagnostic the retinal. So we have tackled 35 retinal diseases, inherited retinal diseases, and sort of constructed a collaborative structure. So here is the National Tokyo Medical Center, National Institute of Sensory Organs. And then these are the 25 departments of ophthalmology. They will provide the patient information, also the blood or saliva samples, and they will come to the Tokyo Medical Center. The DNA will be isolated. The DNA will be sent to RECAN or other facilities for the next generation sequence. It will be sequenced. And then it goes to the National Institute of Genetics, where they have the supercomputers to analyze hundreds or maybe thousands of exome data simultaneously. And they will provide us with a short list of gene lists. And then we will have to decide with these people what the mutation are. Once the mutation is found, we go for the functional analysis, make a knock and mouse. We do iPS cells. We try to find, decide whether this mutation is actually the real cause for the diseases. And then once this is established, we will communicate with these institutions to screen the drugs for the development. This is the scheme that is currently running for 4 years. And now I like to emphasize that we have a system to do a diagnostic. So these are the university in Japan. They will send us the samples of different diseases. However, there is always a diseases leader who will filter. So some universities are very good at Stargardt diagnostics, but it will be not so good, maybe not good in Leber congenital amaurosis. So there will be always a leader for each diseases, and they will check all the registered information to Tokyo Medical Center, and then they will, if it's not good, they will send this doctor that this is not a Stargardt, or this is not a Leber congenital amaurosis. You have to re-diagnostic this again. So this is the filtering, first filtering. Once it passes the filtering, then it comes to me, and then we go for the whole, one patient, the exome sequences. If we don't find a known gene mutation in this process, then we go to the full pedigree exome analysis. And finally, there will be a short list of candidate genes, and the leader and me will decide which mutation this is. And then if it's decided, we will be going back to the patients, and also we go into the functional studies. So to do all this, we have to do a registration system. We have developed a registration system so that we don't have to email each other every day to exchange data. If you go into the website, you will know what the process, how is the process, the situation in your of your samples, whether it's being DNA has been isolated, it has been sequenced, is what stage the samples are. And also you can put in your comments and to here. So this systems was established in Japanese, but April 1st this year, we will totally convert this into English so that AEGC can probably use this systems for patient registration system. So this is all the pull down menu, and then you select the diseases. You can attach ERG, OCT, FA, and some other information in JPEGs. And if you push this button, all the patients and the genetic information will be published into a PDF files, in one PDF files. So you don't have to click and to find the mutation and information for all each patient. Just click one button and you get all the information available. So this is the system that we used, and then we have collected 602 pedigrees and then 1,746 samples together. We have been using not just blood samples but saliva samples. This is very convenient for people who are very elderly, has been already diagnostic, but elderly cannot come to the hospital. We just send this kit to the home, and they can just wash their teeth, wait 30 minutes, take 2 milliliters saliva, push this cap, mix it, it'll be stable for 1 year at the room temperature, and then just put it into the regular mail and send it back to us. So this is very convenient. Now this DNA is extracted by us and then fragmented, and you using the exome capture kit, it will be isolated, and then the exome the sequence will be done by 100 reads in average. And this 100 reads is very important to cover all the exons of the genomes. So how many samples do you need? This is the recessive type. So this is the homozygous, this is the compound heterozygous. Usually we definitely need the patient and also the parents. This triplet is must to identify new genes. But if you have a sibling, a brother or sister with a affected, then it will be mostly you will now narrow down to maybe 2 or 3 genes at most. For the dominant type, however, you need 3 generations. 3 generations, that means 6 samples plus 2 affected in the relatives. So that's 8 samples. It's very costly to do a dominant. So we cannot go for exome sequencing for every pedigree that comes. But that's why we need to establish quickly the database so that you don't have to go for everything, but just selectively go for the mutations. So what kind of mutation we're thinking about? The single nucleotide variants, small deletions, small insertions. These can be covered by exome sequencing. However, there are large deletions, large insertion, inversion, duplication, and chromosomal translocations. And these have to be covered by whole exome sequence, still relatively expensive, $1,600 per person in US dollars. So we would like to cover as much as possible by exomes, and then eventually for the pedigrees that doesn't have the candidates within those exome sequencing, then we move to the whole genome sequencing. But the sequencing prices has been down significantly, and now it's coming to the point of $1,000 per person, which is very, very attractive to us. All this software that is we are using for the exome sequencing is all freeware, so and the computer is available anywhere. So once we have the website going, we'll put all the softwares and how to do the exome studies on the website, so that anybody can set up a computer and start their own exome analysis. So this is the result for the Japanese populations: Leber congenital, occult macular dystrophy, a recessive type of ARP, and this. As you can see, we have quite a bit of large numbers of LCA, and we're finding new genes for this. And also, we have about 250 pedigrees, so we're finding new genes here. But overall, only 17% of the pedigree were identified with the novel genes. So that means there's a lot of more genes to be identified in the Japanese population, and I'm sure this will also happen to the Indian population, Chinese population, Filipinos, Vietnamese, and then Cambodia, and etc., etc. So we need to push this pie to this side, and I want to do this in 5 years' time. So these are the genes that we have been finding for the novel genes, a new mutation in novel genes. Also, and I'd like to end by a new gene that we just submitted. This is a new gene for Leber congenital amaurosis. We found in this large recessive families. It's CCT2 is the causing. It's a compound heterozygous mutation. And then we have did the computer analysis of this mutation, and it looks like it's the whole structure of this chaperone protein is disrupted by this mutations. Also, we have tried to make the eye cup from the iPS cells that come from these patients, and then you can see that control is developing very rapidly. However, the mutant is not developing. So there are some development problems with the eye cup development. So in conclusion, we have a consortium of 25 ophthalmology departments, and we have collected this amount of pedigrees, and we have been doing this short select system to capture the exomes and then do the sequence with a high-sec sequencer. Only 70% of the pedigree were identified with the known genes, and the new genes, the novel genes. We have about 25 novel candidates right now, but have been found, and probably we will find more in the days to come. And then we also made we are making iPS cells bank from each patient that are with the novel genes, and then see what kind of abnormal thing is going on in the cells. And I just mentioned that we just identified and submitted a paper for the CC2 for new gene for LCA. So these are the people who are collaboration, and I'd like to thank all the people and also the funding of this. Now the funding for the medical research in Japan will be united to a system called AMID starting in April 1st. So it will be like an NIH structure that all the medical research, basic or translation, will be under one umbrella. So thank you so much for your attention.